All right, so we need to start another lecture, and this should finish up the nerves. And a lot of the fundamentals you'll get from this lecture will help you to understand the sensory system, which I believe is our next section. And so we need to talk a little bit about how does the body and the nerves respond to stronger stimuli versus weaker stimuli? How does the muscles respond to greater stimuli versus weaker stimuli? How does all that kind of get integrated? Is it a stronger action potential? So these are the kind of things that we're going to try to discuss a little bit. So what is important is to understand there's another type of potential that is different than the action potential, and it's known as degraded potential. This takes place in the cell body and in the dendrites, and ultimately triggers the frequency of action potentials. Where this is determined is in a special area in the axon hillock, usually, where there's lots of sodium channels that go through, they're voltage gated. And if a voltage charge is high enough in millivolts, it'll actually cause the sodium channels to open up in the axon hillock. And in doing so, this will trigger action potentials that will then travel down the axon. And we'll get into more about what a graded potential is. But this picture here is illustrating the differences in the frequency of action potentials that ultimately results in, when there's more action potentials, it results in an increase in neurotransmitters being released either to another nerve or to a muscle or to whatever effector cell is there. And the more action potentials more neurotransmitters that are released due to a higher frequency of action potentials, that results, let's say if it's a muscle cell, in more action potentials in the muscle cell. And in doing so, that allow the calcium to stay out longer, allowing for the contraction cycle to take place. So that's just one example of this. And so we're looking at these two different pictures. It's corresponding to, in this case, a sensory cell, and it's showing you the dendrites. In this case, it's remember, this is a pseudo-unipolar neuron, so it's a little bit different in appearance. The cell body is actually down a little further from the axon terminal in this particular example. And we have a stimulus, some type of response. Maybe it's stretch receptors on the skin. It's just a variety of different possibilities that triggers the graded potential in the afferent neuron in this particular example. And so the graded potential increases. And what does that mean by increases? It means that the inside becomes more positive, more sodium's coming in. And so at the top picture, we see a weak stimulus. So there was a weak stimulus down here. So that brought in sodium that made the dendrites or the cell body, depending on the type of neuron, to go up in millivolts, reaching around negative 20 millivolts. So right in this area, we're at negative 20 millivolts because of the type of stimuli caused some sodium to come in. So we have a depolarization where the graded potential reaches a negative 20 millivolts from the resting membrane of around negative 75. And so when it reaches the trigger zone, which would be down here somewhere where the axon hillock is in this particular example, we have a handful of action potentials. Because remember, an action potential is an all or nothing event, meaning that it'll go up to around 20 millivolts and then the potassium channels will open and come back down and hyperpolarize. And then immediately behind it could be another action potential. And so around 20, so that's the difference, is these action potentials are all the same height. And a small graded potential resulted in a handful of action potentials. So a small graded potential resulted in a handful of action potentials. And I'll get into more images describing a graded potential in a moment. However, let's say there was a strong stimulus, and now we go up from a negative 75 millivolts, the resting membrane, up to nearly 0 millivolts or 5 millivolts. So we've increased the depolarization substantially more in this strong stimulus. We went from a negative 20 millivolts in the weak stimulus up to 0 or 1 millivolt or so. This strong graded potential means that there's lots of sodium that will reach the axon hillock. That is the deciding zone that I've shown you in previous pictures. Now maybe I'll show you another picture. I'll jump to that slide in a moment. But at that point, all these sodium channels open, and then there's lots of action potentials traveling down the axon and real close to each other. So the frequency increases, but the size is the same. See, here we have 20 millivolts. Here we have 20 millivolts. But the number of action potentials dramatically increases that are traveling down the axon. And when it reaches the axon hillock, it'll release more neurotransmitters through exocytosis. But here in this weak stimulus, we have fewer action potentials traveling down to the axon down the axon to the axon terminal, and we have fewer neurotransmitters released. So again, this is what makes the difference between a strong response and a weak response. Fewer action potentials traveling down results in a weaker muscle contraction or whatever the effector might be. But a strong graded potential results in lots of action potentials, which causes a lot more neurotransmitter to be released, and ultimately a stronger muscle contraction or whatever the effect would have been. So we're going to come back to graded potentials in a moment. Realize that neurotransmitters, there's a variety of different types of neurotransmitters. We've mostly talked about acetylcholine. But there's actually a variety of neurotransmitters that do many different things as far as activating action potentials or reducing action potentials, modulating a neuron. So there's lots of different things that neurotransmitters can actually do, both at a local level and if it's a neurohormone, actually at the larger level. And so those are some things to think about. But again, it depends a lot on the receptors that these neurotransmitters are attaching to. So again, we won't go into a lot of detail about these, but here's examples of neurocrines. These could do a variety of things. They'll hook up with different types of receptors, and we'll get into this at the latter part of the lecture, so you may want to come back and refer to this slide later. But we'll talk about what an AMPA and MDA kind of receptors are, but there's GABA receptors and glycine receptors and purines and even nitric oxide can all function to alter the nervous system and its receptors. Some are found in the central nervous system and so forth. So these are major neurocrines. And again, a neurocrine is kind of like a neurohormone. So sometimes, but it's mostly working at a local level. So they would call this a paracrine. So a neurocrine is kind of working at a very local level. It's not necessarily universally traveling around. It may in some cases, but it's mostly regional. So that's why we call it a neurocrine, which is kind of like a hormone that works in a region called a paracrine, but specifically for nervous system. And so we have also, as we've already talked about, acetylcholine. This works on the more well-known receptors like chlorogenic receptors and mesocrinic receptors. And then there's also neuroepinephrine, which works on the adrenals, the androgenic receptors. dopamine, serotonin, and histamine. And you probably might have come across these kind of names before. So again, some of these neurotransmitters, neurocrines, will work on skeletal muscles like acetylcholine or norepinephrine. Will work on smooth muscles. Okay, so now we're going to go back and talk about graded potentials a little bit more. So here's where we first talked about how the graded potential can be different heights, different strengths, and action potentials are kind of an all or nothings, and they almost all look exactly like carbon copies of the previous one. So they're essentially do the same thing really. Start at around negative 75 millivolts, move up to 20 millivolts, come back down into a hyperpolarization of around negative 85. You can see that even in this graph. Or just a little bit lower than the resting membrane, and then it's back to another action potential. But graded potentials, they can be all sorts of different sizes. And where does action potentials take place? In the axon. Where do graded potentials take place? In the dendrites and in the cell body. Okay, so let's go look at what a graded potential is doing. Okay, so just to keep track of things, this is our presynaptic neuron. You can see it over here in this picture where I'm using the pointer. And then over here is the postsynaptic neuron. So anyway, this is the synapse, and neurotransmitters are released into this little region that can either activate an action potential or actually decrease the likelihood of an action potential or modulate the neuron in other ways, depending on the type of neurotransmitter or neurocrine that's released. And kind of, if you understand diffusion, when the neurotransmitters are released, this channel opens up. So the acetylcholine or whatever will attach to this receptor, opening up this channel. Remember, channels are on cell membranes. And then sodium will come in, most likely, if it's opening, because remember, there's more sodium on the outside of the neuron than on the inside. So the sodium will flow in. And so that immediate region, there'll be a spike in sodium, an increase in sodium. And then the sodium will diffuse across the cell body from the point of where the concentration, where it first came in. And so as this like diffusion, you're moving from an area of high concentration to an area of low concentration. And if you look at this picture and look at my cursor where I'm pointing, you'll see that the sodium concentration is stronger where it first comes in and gradually gets weaker as it travels through the cell body. This is the postsynaptic cell because it's after the synapse. So again, the concentration decreases. Now, this can be loaded with axon terminals, causing sodium to come in throughout all different regions. So when this sodium travels through the cell body, it's going to reach the axon hillock. This is the trigger zone. This is the deciding zone on whether or not an action potential will actually occur down the axon. Remember, it's the area where you got the cell body and then it comes down, in the case of the motor neuron, and it's that area where it kind of looks like a cone coming into the axon, that we call that the axon hillock. If you look at my cursor here, looking at the presynaptic neuron in this particular case, this is the axon hillock. And then if you look at the postsynaptic neuron, looking at my cursor again on the screen, you can see this is the axon hillock here. So the sodium is built up in strong concentration here, and it's going to diffuse across the cell body. And if the concentration is strong enough, meaning it's high enough in amount of sodium, if it's high enough, it will turn on, what I mean by turn on, it will open the sodium channels that are loaded in the trigger zone. Because remember, what do we know about sodium channels? that are outside of these receptors, they're voltage gated. And so if the voltage is high enough, those sodium channels will open and trigger an action potential. And remember what the millivolts were? If we go back to this slide here, it was the threshold is around negative 55 millivolts. And if you look at the previous presentations, you'll see that that's what the typical trigger threshold is for opening up those sodium channels. Different authors and cells may be a little bit different, but that's about the extent of it. So if you get to that negative 55 millivolts, and I'm hoping you remember this from the previous lecture, if not, you need to go back and watch it, the activation gate opens, sodium flows in. Then we reach that peak, and then the inactivation gate closes and no more sodium comes in. So if that's confusing to you, you definitely need to go back and review the previous lecture. But remember, graded potential can be all sorts of different variations in millivolts, while an action potential is this replicated negative 55 up to 20 millivolts back down to negative 75, and then ready to rock and roll again into another action potential. So again, if that doesn't make sense, watch some videos, watch my previous videos, try to make sure you understand that. So here we have the sodium going through the cell body. And so that's what we see, excuse me, that's what you're seeing in this picture here. You can see that there was an action potential that traveled down this axon. The neurotransmitters were released across the synapse. We had a strong threshold that traveled through the cell body. And if it remained high enough, we have an action potential. If it isn't strong enough, there's no action potential. So let's look at these two examples. On the left, you see a graded potential that was pretty strong initially. Maybe it was just, but remember, it's based on frequency. So maybe there's just not as many action potentials traveling down the axon. And so when it reached the axon terminal, in this potential example, a few neurotransmitters were released. That opened up, let's say acetylcholine. It opens up or allows sodium to come in. It's excitatory is what we call that, meaning it's depolarizing. And so sodium comes in and then it flows through the cell body. And as it travels through the cell body, again, it's a strong concentration initially, but in this particular example, the cell body is pretty long in this, microscopically speaking, or some other reason, or maybe this wasn't a very strong excitatory response, meaning there wasn't a lot of neurotransmitters released. But regardless, when the sodium finally reaches, and finally is again milliseconds, it's so finally in quotes, but when it reaches the trigger zone, and again, the trigger zone is the axon hillock with lots of sodium, voltage-gated sodium channels. When it reached there, it was below the threshold. Again, the threshold is around negative 55 millivolts. And so we don't have an action potential travel down the rest of the axon. So it wasn't strong enough. Even though this was excitatory, the graded potential, by the time it reached the trigger zone, it was below that negative 55 millivolts. So the sodium channels that are present there did not open. The activation gate did not open. Again, go back and look at the previous lecture if that's confusing to you. Now, on the example on the right, the stimulus was stronger. That meant more neurotransmitters were probably released, causing more sodium to come in. Those are some of the reasons. There's other reasons for this too that I'll get into as we continue in this lecture. But irrespectively, even though they had similar concentrations initially, the concentration stayed strong enough. Maybe it lasted longer. Maybe the neuron was actually smaller, so it stayed more concentrated. There's lots of different possibilities. But what you're seeing in this picture on the right, even though the degraded potentials were similar initially, the concentration stayed stronger throughout traveling, in this particular example, stayed stronger traveling through the cell body. Now, if you look at my cursor, you can see that's represented by a bigger arrow. And if you look at the right, you'll see that the voltage stayed higher, so that when it actually reached the trigger zone, and that's what these needles are representing, reading on oscilloscope or whatever they would call it, that when it reached the trigger zone, those voltage-gated sodium channels, activation gates, opened. And when they opened, because they were over negative 55 millivolts, the threshold, action potentials were triggered to travel down the axon. And if it's really strong, you'll have more frequencies of action potentials traveling down the axon, like you saw in this previous picture right here. A really strong threshold results in loss of action potentials on the lower portion. Weak graded potentials, fewer action potentials, or none, as shown in that previous picture. So again, looking at the sodium traveling through, if the sodium concentrations are high, you're going to have lots of action potentials traveling down the neuron. If the graded potentials are weak, you'll have fewer action potentials at the trigger zone, or you'll have none. So that's how the cell, in a sense, integrates that information. So again, here's the trigger zone, the axon hillock. It's the deciding zone. And again, what makes it so this way is the fact that it has so many, and I don't know what the numbers would be, hundreds, thousands, I'm not really sure, but there's lots of voltage-gated sodium channels present in this trigger zone. And if the sodium concentration is over negative 55 millivolts, those voltage-gated channels will open, triggering the sodium channels down the axon to open in sequence. And then, of course, you should already be familiar what happens during an action potential. And I'm not go back and check out those other lectures. So just realize this information is traveling down these neurons. So here you're going to have lots of axon hillocks surrounding a neuron. And each one of them is talking to this nerve. They're converging onto this nerve. And in this case, if they're all excitatory, it's probably going to make the concentration strong enough to trigger action potentials. Now, remember how I talked about distance as well? Well, we have 1, 2, 3, 4. They're numbered that way anyway. Realize that imagine if sodium started coming in right here on number 4. The concentration doesn't need to be as high to trigger an action potential because the diffusion is much shorter. So that concentration is going to be pretty strong already, and it's going to be right next to the axon hillock, triggering the action potentials. Now here on the number 1, it's quite a bit further. So imagine diffusion taking place as my cursor is traveling through. It's so much further away that it either has to be really strong to begin with, or it might not be strong enough and it reaches that axon hillock. So you can see how distance does make a difference because we're talking about diffusion across this cell body. So even going back to this picture, it's not in this picture, but imagine if you had another, um, you had another axon terminal right here that's not shown as, like this is an axon terminal. Imagine there was one right here, and you had that strength of sodium coming in right there. If it was right next to the trigger zone, well, heck, it's probably going to trigger an action potential because the concentration takes a while to weaken. Again, it's diffusion through the cell body. So there's a couple other terms that kind of represents this. Some are known as spatial summation, and others are known as temporal. Spatial is, of course, where are these neurons located, and temporal is the timing of action potentials. And this is in reference to the presynaptic and the postsynaptic neurons and how they interact with one another before they reach the axon hillock. The next pictures will help you to understand this. So here's an example of an additive summation. Additive meaning that obviously they're excitatory, they're added together to make a strong response. And so here we have a couple presynaptic neurons, because again, they're before this synapse. Here's our postsynaptic neuron. It's called convergent because we have all these different neurons coming to it. And here we have a synapse, of course. So this is one's actually pretty close to the trigger zone. So it's going to have probably a stronger influence on this axon hillock, this trigger zone. But in this particular case, all of them are excitatory. They're all increasing the likelihood of an action potential. Okay, so anyway, so you're seeing what we're looking here is an excitatory response from these three different axon terminals, all of them increasing sodium. And so what happens is together, their concentration stays strong enough that when they reach the trigger zone, it stays above the threshold, causing action potentials to take place. However, maybe if only one was turned on and these weren't happening, maybe it wouldn't have been strong enough. It's hard to say because, again, we don't know really what's happening in all these different presynaptic neurons. This one being closer has the most influence on the trigger zone because of the diffusion will be the strongest there when it reaches the axon hillock. So again, these are a lot of hypothetical possibilities. But again, the reason why they're calling it spatial is because it's spread out, spatial. And then it's a summation because they're all additive. And so it's in this case, it's an additive summation because they're all excitatory. But let's say some are inhibitory, even though it's spatial, you might actually have an inhibitory summation. And so that's what we're seeing in this spatial summation here. This is an example of an inhibitory summation. In this particular case, we have two excitatory, but notice we have a negative one over here. So what is making these positive? Well, sodium's coming in because, again, maybe an acetylcholine or something attached to a receptor, opening up the sodium channel, sodium flowed in, making it more excitatory. But in this particular case, this neuron, the inhibitory neuron, actually reduced the amount of sodium coming in. Well, it reduced the voltage. I should point it, make it more clear. The sodium increased the voltage, and in this case, the inhibitory neuron was actually reduced the voltage, just to make it very clear. So what could have happened? Well, I'm not sure exactly. And maybe this caused potassium to leave, or maybe this caused chlorine to enter, but whatever the reason, the inside became more negative. And in doing so, when it reached the trigger zone, the millivolts were below negative 55. And so we did not hit the threshold, and thus there was no action potentials, or there are fewer action potentials depending on the scenario. So that is an example of inhibitory summation. Now, temporal can also be combined with spatial, but temporal is referring to time. So in this case, we're just looking at one neuron, but it could be multiple neurons still involved. But in this case, we have a neuron who has action potentials that are pretty spread out. And so they're traveling down the axon pretty far apart. And so when it reaches the axon terminal, perhaps the action potentials are pretty weak. So there's fewer neurotransmitters released. But irrespective, they're opening up the receptors pretty far apart in time. So when the diffusion takes place through the cell membrane, again, that's the degraded potential we're talking about, that when it reaches the trigger zone, the axon hillock, it is well below the threshold. So again, now let's look at this figure on the right. This is showing you spatial summation, temporal summation, but in this case, the summations are much more close to each other in time. So, because you see down here at the bottom, it says time in milliseconds. And so when the two come together, the diffusion is, basically you have a little diffusion going down and another one right behind it. And so when they get to the trigger zone, they're actually above threshold. And when they're above threshold, it causes all the sodium channels in the axon hillock to open up and send that action potential down the axon. And that's what we call spatial summation. It kind of reminds you of the twitch summation, if you remember from the muscle lecture, where you had the two twitches close to each other and it resulted in a big twitch. But in this case, it's the two sodiums that are diffusing through the cell body coming together. And when they reached a threshold, they're a little bit stronger concentration than they would have been had they been spread out. And this one diffuses first, then this one diffuses. Now remember, this is, again, in time. That's what down here on the x-axis, you see it's in seconds. Remember, though, temporal summation and spatial summation, all these different factors are happening at the same time. So it's a very complex mix. And you can literally have hundreds or thousands, I don't know for sure. I don't know if it's that high, but you'll have lots of axon terminals synapsing onto the cell body. And so that's what this picture is showing you here. This is the cell body, and then look at all these different axon terminals. So they're all coming from the presynaptic neuron up here, synapsing on the postsynaptic neuron. They're all either excitatory or inhibitory or doing all sorts of different things to this degraded potential, that when it reaches the axon hillock, which you can see is kind of buried under some more axon terminals, it can turn on those sodium channels and thus triggering action potentials down the axon. And so that's what we're seeing here. So these ones are going to have probably a greater influence on action potentials than those up here, just because of the sheer distance those diffusions take place and travel. So how do we talk about in regards to neurons talking to other neurons? Some are known as divergence and some are known as convergence. And this is based on the nerve networks. And again, this is going to be very complex. But essentially, in the case of divergence, one neuron is talking to many different neurons through their axon terminals. So they'll have an axon, and some of their axon terminals will synapse on this neuron or this neuron or that neuron. And then this neuron's synapsing on a variety of neurons. And so it essentially allows this neuron to kind of be a master neuron over multiple neurons. It's kind of like almost like a top-down control in a business model or something of that nature, or bureaucracy. Here we have the commander in chief, and it's and telling the middle managers what to do, and then they go off and tell these neurons what to do. So again, this is a divergent pathway where one neuron is talking to many neurons, and so has a strong influence on a variety of neurons. While a convergent pathway is multiple neurons are converging onto in this case, this picture shows a single neuron. But you get the gist is that many neurons are coming and converging onto one. And so in a sense, this one neuron is deciding how to respond in regards to many stimuli coming to it. Might be the surface of skin, for instance, and it's all coming down to a neuron that then will send a signal to your brain. This is one of the reasons why some areas of your body is more sensitive than others in regards to its receptive area. But there's lots of reasons for having a convergent pathway. Basically, lots of presynaptic neurons are converging onto one postsynaptic neuron in that particular example. The synapse itself can actually be modulated as well. We can have presynaptic inhibition as well as postsynaptic inhibition. I guess you could also have presynaptic excitatory responses and postsynaptic excitatory responses as well. But these are examples of synaptic modulations that can inhibit a response. So here we have a presynaptic inhibition taking place where we have an excitatory neuron coming in, exciting an action potential to travel down this neuron in the postsynaptic nerve. And then right before the synapse, we have another neuron coming in that actually inhibits the action potential and the release of the neurotransmitters. So this makes the inside of this neuron more negative at the terminal, at the axon terminal, so that the calcium doesn't come in and cause exocytosis and the release of the neurotransmitters. So these other axon terminals still release neurotransmitters, but this one doesn't. So we call that presynaptic inhibition. Excitatory, as you can see this flowchart, excitatory neuron action potential is generated, and then an inhibitory neuron blocks it. In some cases, maybe there would be an excitatory one too. This is sometimes seen in regards to pain receptors. We'll see some type of presynaptic inhibition. Here we have postsynaptic inhibition, which goes along with the idea of the summation that is inhibitory summation that you saw in the previous slides. Here we have an excitatory neuron and an inhibitory neuron. This inhibitory neuron thus makes the inside more negative. And so when it reaches the trigger zone at number 3, it's below threshold, below that negative 55 millivolts. And so those sodium channels don't open up and we don't have action potentials traveling down the neuron. So this is an example, again, related to what we had already seen, but taking place at the cell body. Now let's talk about the different types of receptors that are found with the postsynaptic neuron. So here we have our presynaptic neuron. You can see my cursor here. You can see that the neurotransmitters are inside the vesicles, and then they're about to be released if a action potential travels down. The neurotransmitters are released in the synapse, and then they will attach to the receptors They're found on the postsynaptic cell or neuron. And so when the neurotransmitter comes along, it'll attach. Some are going to be a fast response and some are going to be a slow response. Some are going to be excitatory and some are going to be inhibitory. If it's acetylcholine, for instance, it's going to be excitatory. It will attach the little red. Imagine if that little red was acetylcholine. It will attach to the receptor. And then this receptor door will open and sodium will come in, and then we'll have an excitatory depolarization. So again, this would be what we call an inotropic neurotransmitter. And then we also have what you call a metatropic receptor. And this is what happens when you have what you call a G protein linked receptor. Maybe I'll find a video for your homework to help you to appreciate this a little bit more. But in this case, a neurotransmitter attaches to this, what they call a G protein linked receptor. This is the G protein. This is the receptor. The neurotransmitter attaches, and then it activates this G protein. So just realize that this is one of the ways that cells works. And so through a complex chemical cascade, that's what it's basically, when I say chemical cascade, that means that there's enzymes responding and chemistry going on inside the cell. So that's what it's saying here, activated secondary messenger pathway that then can modify proteins or synthesize proteins, maybe through going through the DNA and messenger RNA synthesis, but can actually change the neuron in such a way that it coordinates intracellular responses. So that is kind of a long and slow process. And this is something that you see more maybe in the formation of memory because it modifies neurons. Now, often they can be linked with an ion channel as well. And if so, it can either excite or inhibit again. So let's talk about that. So let's imagine that, let's go back to this chemically gated ion channel. Imagine a neurotransmitter like acetylcholine came in. If it opens, it's an ion channel, and if more sodium comes in, it's excitatory. Now let's imagine it's a different type of neurotransmitter on a different type of channel receptor. But in this case, it opens up and potassium leaves. So it's a potassium-related channel. And what happens when potassium leaves the cell? It becomes more negative on the inside. It hyperpolarizes. And when it hyperpolarizes, you actually inhibit an action potential from occurring. So it's inhibitory. Or let's imagine chlorine coming in. If chlorine comes inside the cell, the inside of the cell becomes more negative. Again, inhibiting action potentials because the inside becomes hyperpolarized. Because remember, it's all about the thresholds and so forth as it travels through the cell body. Again, this could happen in other places, but in this case, we're talking about the dendrites and the synapse and so forth. And then again, a G-related protein can stimulate a wide range of chemicals that can also trigger the ion channel if it's associated with it closely. If it closes, if the ion channel closes and sodium doesn't get to come in, so maybe there's some kind of response where it either attaches here or attaches to this receptor, but either way that those channels close and stay closed and sodium can't come in, what happens? The inside stays negative, so that would make it inhibitory. Less sodium coming in makes it more inhibitory. Less potassium leaving, if that channel closed and was a potassium channel, so the potassium doesn't leave, the inside stays positive. And if the inside stays positive and sodium comes in, maybe for this other channel, it becomes more excitatory. So these are all the different ways these neurotransmitters can kind of influence whether or not something's excitatory or inhibitory. But as a general rule, if sodium's flowing in, it's excitatory. If potassium's leaving, it's inhibitory. If sodium can't come in, that channel is closed, and sodium can't come in, it's inhibitory. And if potassium can't leave, it's excitatory. So just make sure you get that. If sodium's coming in, it's excitatory. If sodium can't come in, it's inhibitory. If potassium can leave, it's inhibitory. If potassium stays in, it's excitatory. And all these different neurotransmitters can influence that, as well as this secondary messenger pathway can influence these channels. And again, you see this more in how synapses form and connections form between neurons. And we call that long-term potentiation. Long-term potentiation is when a neuron is interacting with the presynaptic neurons interacting with the postsynaptic neuron in a way that makes a stronger connection between those neurons, increasing the communication between them. And this is where we start to think memories are formed, long-term memories and connections, or if you learn a dance routine, you're strengthening neural pathways that are utilized a lot. And so glutamate is released, and that glutamate can influence what we call long-term potentiation. What happens is it attaches the sodium receptor channels. Sodium comes in, that would make it excitatory. And it also attaches to this calcium channel that allows calcium to come in as an MDA receptor. And I don't know a lot about these different receptors that magnesium leaves. But then there's this complex chemical pathway that takes place. So it's kind of like the G-related protein that we saw in the previous one. But either way, realize that there is a chemical cascade that takes place, and we call that secondary messenger pathways, with this calcium flowing in, that changes the neuron. Different proteins might be formed or so forth. But ultimately, it might cause the release of paracrines that travel back to the presynaptic neuron. So remember, a paracrine is like a hormone. And again, this is a very simple, wide version of what's going on. But a paracrine is like a hormone, where, but it's only within a short little small region. So when the neurons talking to each other, the presynaptic and the postsynaptic, that paracrine release might help modulate this neuron, and kind of they start forming this stronger connection between the two. And again, I know that's oversimplified for what's happening, but we believe this is some of the things that take place in forming strong connections between neurons that could be related to memories or learning a pattern or speech or whatever memory-based activity that takes place. And again, what's happening is this glutamate's attaching, causing sodium to come in and calcium to come in, causing a secondary pathway that causes the release of paracrines that come back, and they just start forming a stronger connection between these two cells. So again, this may be key to learning and memory. Glutamate functions as an iotropic and metatropic. Remember, inotropic means ions, like sodium, calcium, whatever. Metatropic means it's all the secondary pathways that take place that ultimately cause the synthesis of proteins or and paracrines that are a little maybe amino acid chain that can then travel back to the presynaptic neuron and form this stronger connection. I might see if I can find a video for you to watch for your homework to get that a little bit better. But anyway, that is the idea about this long-term potentiation. And I think I will add that to your homework so you can get an idea about that. So this will conclude this particular lecture and finish up the neurons. We will move into sensory. How does the eyes work? How does taste receptors work in our next set of lectures? So basically, you need to do, again, a summary that's a little bit more than half a page. And I would like for you to explain graded potentials versus action potentials. Try to explain how a graded potential and an action, compare and contrast a graded potential with an action potential, and explain what's happening maybe in this picture here. So draw a little diagram of what's happening in this picture. Well, let's make it this picture here. Let's make it a picture based on this. Add this diagram and explain what's happening in regards, and it doesn't have to be perfect, but just, again, explain, after you kind of give your summary, what is a graded potential, get into what the trigger zone's doing, and so forth. I hope you get the gist from this lecture. If not, don't hesitate to look at your textbook or look up something on the internet. Anyway, take care and good night, or goodbye, or whatever, depending on when you watch your presentation or this lecture. Take care.